Hello and welcome. Good morning to today's class. I'm hoping that you're all having a good morning so far. It's Wednesday. Bit of a rainy, thundery old night here in Ottawa last night. Not the world's most pleasant thing to sleep through, but things are looking pretty bright this morning. So grab your coffee, kick back and relax. I guess you could always listen to this while you're driving to work or running. Maybe you shouldn't listen to while you're driving, but you can kind of do anything. But it seems like a nice thing to listen to over a plate of eggs and a nice cup of coffee. Anyway, so we're going to talk today about DNA sequencing and other methods. And I think it's safe to say that my goal in this class is not to turn you into dedicated methodologists. My goal is to teach you about the genetics of mental health. However, I think it's valuable to have some knowledge of the methods that are used to carry out that research, because if you're looking at papers in the literature or if you're trying to understand my slides, you probably should understand roughly where the data come from. And of course, if you're interested in going on to do the work yourself, knowing where it came from is a big advantage. So with that in mind, we're gonna take our only main class on sort of the molecular biology and the sort of technical methods used to make these analyses, and anything else that comes along we'll address in due time. So let's get started. Here's the title: DNA sequencing and other methods. So we can begin by asking, what does a researcher in this field need to know? If you're, I guess you can kind of divide the work that we're curious about into two broad categories. There's questions of what are the genetic correlates of mental health and neurological disease? What genotype gives you a higher risk of this or that? And then there's a second sort of question of what epigenetic marks might be related to these things. Whether those are tissue-specific epigenetic marks or kind of epigenetic marks in surrogate or peripheral tissue, that's somewhat of a separate question. Most of what, obviously for the genetics of mental health, most of what a researcher needs to know is the genotype of a lot of people who either have or don't have a given mental health condition or a given neurological disorder, whatever the case may be. So most of the studies that we are going to look at here in this class are going to look across the entire genome. They're called genome-wide studies, and they involve genotyping a large number of samples across the entire genome. You're trying to capture a fairly representative swath of human variation, and then among the range or within the range of variation you detect, you want to see if certain variants are more common in your cases or your controls, and draw conclusions from that. So to begin, researchers need to know about the full spectrum of variation in the population they're studying. Now, I suppose this is maybe a little bit obvious, but how are you supposed to study the genetics of mental health if you don't know what the range of variation is? You could look everywhere, but that wouldn't be very efficient. What you need to do is figure out where to look where there's actually differences between people. As I mentioned in the last class, humans are, 99.9% identical across their entire genome. A very small fraction of individual genomes are even different between people. You need to look at places where there's differences before you could ever hope to find the correlates of mental illness or whatever it is you're curious about. Now, we will learn in subsequent lectures about concepts like linkage disequilibrium and the role that allele frequency plays in this. Briefly, the concept of linkage disequilibrium allows researchers to sample a fairly, a comparatively small section or, slice of the genome and still get a very accurate sense of the overall level of variation that individuals have, only because variants are in LD with each other, and you can genotype one variant and you'll know with a fairly high level of confidence what the genotype is for all of the other variants that are in linkage disequilibrium with that one that you genotyped. That's putting it briefly. We'll get into that later on. So the genome-wide studies that we're going to be looking at tend to use microarrays, as we'll discuss, that study SNPs or variants that have a high level of individual variation and don't include SNPs that are in high linkage disequilibrium with those. There's no point in measuring or going to the trouble of measuring two SNPs that are in LD with each other because they're always going to be the same, or they're always going to travel in lockstep. So this is the kind of stuff that researchers need to know. Epigenetics studies or epigenomic studies, in contrast, it's a little bit tougher to choose areas of the genome that you want to focus on. You're much better off just working with the whole genome and seeing from the data where the interesting parts are. So if we're talking about genotyping and doing genetics studies, you need to know, you need to have a set of participants or subjects, and you need to know their genotypes, because how else are you supposed to do the study? Now, the kind of classic behavioral genetics studies that you would see in psychology departments or in kind of older-fashioned molecular biology departments, or really any facility where the ability to do a genome-wide study is restricted by lack of access, or just the study design doesn't favor it. You would use sort of candidate gene studies. This is what you might call hypothesis-driven research, because in a candidate gene study, you have a specific area or locus that you're curious about where there's variation between people. In all likelihood, it's in a gene or located near a gene that you already know is relevant. Think about the what I was talking about, biochemical disruptions to known biochemical pathways like the COMT-val-MET allele, for example. You have a gene and you know what it does, and you know about a variant that you have a hypothesis about. Having it is worse than not having it, that kind of thing. And so you can form a hypothesis and design an experiment that looks just at that particular gene. With these kinds of studies, you can have a much smaller sample size because the range of outcomes you're testing is much smaller. So the advantage to a candidate gene study in this sense is that you can design in a more elaborate experiment. You can bring people in, you can ask them about you can do psychological experiments on them. You can do like social stress tests. This Trier social stress test is like a public speaking test. You can do all kinds of different weird and wild things to test their behavior, to test various aspects of their phenotype. And because you have a smaller sample size, this allows you to invest more time and resources into just collecting information about the sample. And then because you're only looking at one area, or maybe a small handful of areas, the multiple testing burden is much reduced. Now, has anyone taken statistics yet? If you have, in all likelihood, you're familiar with this concept of repeated measures and the need for multiple testing correction. And that is a very relevant fact for genomics, because essentially, in a genome-wide study, you're looking at hundreds of thousands of different alleles, and for every allele, you're doing a test to see whether it is related to the phenotype or the trait or the illness that you're looking at. So you've got hundreds of thousands, if not millions, of individual tests being performed across the genome. And if you have a cutoff for statistical significance of p less than 0.05, you're going to get zillions of significant results just because there's a certain likelihood that the data will come out significant just by chance. 5%, well, anyway, I don't want to get into too much about what the p-value actually means, but the point of the matter is that the more places you test, the more you have to, the higher the standard has to be for considering evidence significant. Okay, but if you're only looking at one area, that issue isn't there. You can have your p-value cutoff of 0.05, and it's actually fairly easy to detect a difference if you're only looking at one place, or at least it's if you've chosen a good place. The standard is much lower. So this is kind of the advantage of genotyping on a small scale and doing your study that way. This is also used, I should say, in clinical approaches. If you're looking, if you're looking to see if somebody has a given genetic risk factor, typically that specific allele would be genotyped on its own, and you wouldn't do a genome-wide analysis of somebody in a clinical setting. You would just look for a specific gene or a specific allele and see whether or not they had it. So I put this in. I don't think I don't really want to get into how PCR works, polymerase chain reaction. I would hope that at this point you've at least had some contact with this, either in your wet labs or your biology classes, or from wherever else, but it's not, you know, you you need a bit of a review. I put this video here. There's a link to it in the slide, or you can just type this into YouTube and it'll come up. It's by this channel called the Amoeba Sisters, and they make these really sort of cute sort of videos about biology that are very very nice high production values that explain in a nice clear way how different things work. Actually, if you're looking for a primer on basic genetics and other aspects of biochemistry and molecular biology, they've got lots of good content in that department as well. But I would like you to take some time and either learn or refresh your memory about how PCR works, because this technique is at the very center of really all molecular biology and all of the things we do here. So even if I don't mention it as part of this, as one of the steps in the things I'm talking about, it's in there in the background. There's some step that involves it. So you really ought to know how it works just for your own sake. Now, one thing that I think is kind of related to that, but that I do want to emphasize because it's very central to everything we learn about in terms of methods, is this idea of hybridization. So the magic to DNA really is that you have two strands of material that are complementary to each other. And through the magic of base pairing, these complementary strands will bind and form the classic DNA double helix. This tendency to have a strand on one side and its reverse complement on the other is what enables DNA to be copied. It's what enables you to repair DNA. It's what lets DNA do all of its magic. And the basic principle of it is that a given sequence or a given strand, a given single strand of DNA will bind quite readily to its reverse complement, and they'll stick together. And this is a general purpose aspect of chemistry. You don't need to know anything about the hydrogen bonding of purines and pyrimidines or any of that kind of stuff. You just need to know that complementary letters will tend to bind to each other, and the more of them that are bound together, the stronger of a bond it tends to be. Also, the more G's and C's there are, the stronger a bond it tends to be. So this is, a very basic aspect of DNA, but the beauty of it is that it can be applied or can be exploited for many different kinds of lab techniques. What you would typically see in research is that researchers would design probes. They can call different things, but typically you might call them a probe. And they are simply short stretches of synthetic DNA that are complementary to areas of interest in the genome. So, for example, if your target sequence is this, CGA, TC, GGT, the reverse complement to it that would bind perfectly with it is this sequence, and every letter binds with its mate in the opposite strand. And this is how base pairing works in nature, and this is how DNA is able to be copied. But you can also design these probes, and then when you expose them to DNA in your test sample, the probes can bind to places on the genome that they match up with by base pairing. So this is called hybridization, the tendency for these probes to stick to complementary regions of the genome or of the sample that you've given it. And this basic tendency really underlies everything that matters about molecular biology, because these probes can be synthesized. You can attach all kinds of other molecules to them and achieve a whole range of different effects. So part of the appeal, I think, I just mentioned this here, is that this is about the only chemical concept you would really need to know to be fairly successful in molecular biology. The DNA pieces fit together kind of like Lego. They join on the principle of base pairing. The longer the sequences are that are joined together, the tighter the bond. The more G's and C's, the higher GC percentage there is in the sequence, the stronger the bond. And the more bases that match up, the stronger the bond. If you can actually have pretty nice base pairing even if one base doesn't quite match, it just is loose. But the strength of the bond depends on how well the bases match up. So all of this stuff is very kind of elementary. You really don't need to know the chemistry of it in detail at all. Yet it gives you so much power in doing these research into genetics and genomics and epigenetics and molecular biology in general. So let's look at some examples of techniques that use this principle. The first one has the word hybridization right in the name. It's called in situ hybridization. So what you're doing in this technique, in situ means like in the actual tissue or in the actual specimen that you're interested in. The principle of in situ hybridization is that you take synthetic DNA probes that are complementary to a stretch of DNA or RNA that you're interested in, and you put them in, you mix them in with your sample, and they're going to float around, and eventually they will bind to wherever it is they match. And then because these probes are fluorescently labeled, you can look at them under a microscope and see physically where they are. Where they've located themselves tells you where in the sample that particular piece of DNA happens to be. So as an example, you can use this to map genes onto chromosomes. If you have a fluorescent probe, say for a given gene, I don't know, sulf or something, and it's stained with a green fluorophore, you can mix it in with some chromosomes that are in metaphase, and it'll bind somewhere, and then wherever you see it binding on the chromosome tells you where that gene is located. You can also use it to label mRNAs, and so fluorescent in situ hybridization, or FISH as it's often called, can be used to quantify gene expression in tissue. So here's an example where a given type of neuron is labeled by this probe. It's a mRNA probe coupled with a red fluorescent dye. I don't know what the probe is for, but it's some kind of whatever, that labels particular cells. And then there's a second probe with a differently colored fluorophore labeled green. And so in this scenario, every one of these green points is a messenger RNA molecule for a given gene. And you can simply go around the cells, and for every cell, you can count up the number of points you see. And that gives you a reasonably good sense of how much that gene is being expressed in every individual cell. So I've got a couple of questions coming up on the chat, so let me pop over to those. GC is stronger than AT because it has more hydrogen bonds. That's correct. Yeah. So the GC has 3 hydrogen bonds, whereas AT only has 2. And actually, so you can kind of look at the GC content of different types of organisms, and it actually gives you a sense of what their lifestyle is like. So for ordinary terrestrial mammals, it's about 50 or 60% or something like that. But if you're looking at extremophiles, like those bacteria that live next to hydrothermal vents where they live in extremely high levels of heat, the GC content of their genomes is very, very high, 80% or more. So in order to keep their DNA from basically melting apart, they have a lot more G's and C's in there holding everything together with the tighter amount of hydrogen bonds. And then the Kiana asks, does it matter if the probes are in reverse versus forward direction? Not exactly. You would just design a probe that's complementary to whatever you're interested in. So when it comes to labeling DNA, it wouldn't particularly matter whether you labeled the forward strand or the reverse strand. You would just design a probe that was complementary to one or the other. Obviously, for every molecule of DNA, you have both, so it doesn't doesn't particularly matter. And then for mRNA, you would have to make sure that it was complementary to the transcript. So you would have to make sure that it matched what the actual transcript is, and of course the transcript is on the opposite strand of the DNA. So you just have to manage that, but beyond that, the directionality is quite arbitrary. Of course, there's PCR. PCR uses primers that are oligonucleotides. They're oligo means few and nucleotide means nucleotide. So they're primers that bind to specific parts of the DNA that you're interested in and begin the process of amplification. You can also use fluorescently labeled probes to quantitate the amount of a target DNA that's generated by each amplification cycle. So this is actually how this is one of the different ways that the COVID PCR tests work, is they have probes that are designed to bind to parts of the COVID genome, and every cycle of PCR amplification, they get a chance to bind. And whenever they bind, they fluoresce. And so the amount of fluorescent signal you get tells you how much binding there is, and thus how much of the product is being generated. You can quantify that. Next generation sequencing is the topic, or the main kind of thing we're covering today. This is this uses hybridization in the sequencing adapters. These are synthetic oligonucleotides that you attach to your chopped up target or your chopped up sample DNA that enable the pieces of DNA to stick to the flow cell, and there's other parts of it as well that help you purify target DNA sometimes, and so on and so forth. But anyway, so hybridization is used a fair bit in next-gen sequencing as well. And then microarrays. Microarrays are kind of old technology at this stage, but they remain the gold standard or the main method used for genome-wide association studies. And here, you have a bunch of probes that are complementary to SNPs that you're interested in. You can have millions of probes that are, stretches of DNA where the SNP is kind of in the middle of them, and the rest of the DNA is complementary to the SNP and the area that's flanking it. And when you take your sample DNA and expose it to the microarray, anywhere where you have a SNP that matches what's on the microarray, the DNA will bind, and you'll get fluorescence, and you can tell whether or not that SNP is present. So this concept of hybridization is really quite universal in all of these methods. You just take as a starting point the fact that matching complementary DNA strands will stick together, and you can synthesize complementary DNA strands to anything you're curious about and go from there. The possibilities are quite limitless. So I want to tell you a little bit about genotyping on a small scale. This is the thing I was mentioning before about these hypothesis-driven studies where you're just genotyping a small number of areas and doing a kind of more elaborate test to compare the different sample groups. So single loci, or single locations in the genome, I guess single loci is kind of a bad way of phrasing it, right? It should just be single locations. Loci is plural of location, right? So, yeah. Okay, so I'll see if I can remember to change that. But anyway, single locations can be genotyped using PCR, polymerase chain reaction. And without getting too much into the technical details, the principle is basically this. PCR will grab a section of the DNA that you're curious about in your sample and amplify it. And with every cycle of amplification, the amount of that target sequence is expected to double. And in this case, the only reason to do this is so that you have enough of it to actually analyze. You want to, if you just put in raw sample, there's not going to be many copies, even if you have a lot of DNA. But if you keep amplifying the area that you're curious about, eventually it'll be, you'll have billions and billions of copies of it, and you'll be able to test whether it's, what it looks like quite easily. So TACMAN PCR genotyping uses this, where for every cycle of PCR, you've got two different probes that are floating around in the reaction. And the probes are complementary to the two different alleles of the particular variant or SNP that you're curious about. One probe is complementary, let's say here, to the G allele, and the other probe is complementary to the A allele. And the probes are labeled with different colors of fluorophores. One is labeled in, I guess, yellow. Well, no, FAM is fluorescein. So one is labeled green, let's say, and one is labeled in red. Okay, so what you do is you run through a cycle of PCR. When the DNA is denatured, you'll have one strand of DNA that's you'll have single-stranded DNA, and then either of both probes are going to kind of compete to see which gets to bind to your target sequence, to this single-stranded piece. And the probe that has a better fit is the one that's going to succeed in binding. So if you have the C allele, then the probe with the G the probe with the G in this position is going to be the one that binds. If you have the T allele, then the probe with the A in this position is going to bind. It's going to be one of the two. They'll kind of fight it out and compete, and the one with the stronger affinity ends up winning. When they bind, these fluorophores emit fluorescence, and one, of course, the A allele, if it binds, it emits a green signal, and if the G allele binds, it emits a red signal. And so you can do this a number of times to build up enough DNA to actually test. And just by looking at the ratio of green to red in your sample, you can tell whether that person has the G allele or the A allele. Do you see what I'm saying? So it's quite a wonderful principle. Let me just pop back into the chat here. I'm sorry, wrong window. So are these different techniques going to be referred to later, like PCR? Yes, that's going to be part of it. I guess you might be asking if I'm going to test you on how PCR works. No, I'm not going to make you. I'm not going to make you like walk through the different steps of PCR. I think that's not something that would be really worth testing at this point. But I think knowing how it works is going to be fairly essential. I think to just kind of getting the methods that are talked about in this course, because it's so fundamental to everything else in molecular biology. So I'm not going to test you on that directly, but like your ability to understand the methods that I'm talking about in this lecture, as well as the other ones that are coming up, kind of hinges on how well you understand these, PCR and other kinds of methods like that. So it's worth learning about for that reason. So when you're looking at the data from this type of PCR genotyping approach, it's very simple to actually tell what genotype your subjects have. You would simply plot the fluorescence signal of the two probes, the green probe for the A allele and the red probe for the G allele on the Y and X axes respectively. And you can and the individuals that you're genotyping will cluster into these specific points or groups of points based on their genotype. So, for example, if somebody has the A allele, and in other words, the T allele, T is complementary to A, what you'll see is they'll cluster up here, where the level of fluorescence on the A channel, or on the fluorescein channel, the green channel, is very high, and the amount on the red channel is very low. If they're homozygous for the G allele and they have CC, then they'll be very high on the red channel but very low on the green channel. And if they're heterozygous and they have one of each, they'll have equal amounts. And so they'll kind of be here on this diagonal point where they have high amounts of green and also high amounts of red. You see what I'm saying? So you can kind of run through this and tell very quickly what the genotype is for a lot of different people just by running these kinds of assays. You're essentially pitting the two fluorophores against each other and seeing which one comes out stronger. So that's how genotyping on a smaller scale works. And this is the kind of thing that you can do very easily in any of the labs in Carleton's neuroscience department. Everyone has access to qPCR machines that are capable of doing these measurements. DNA extraction from subjects is quite simple, so it's a very, it's a very tractable thing to do if you have a specific, allele that you're curious about and you want to do that kind of study. It also, I should point out, is used for genotyping like transgenic mice and so forth. You can tell what the genotype of those guys is by using this kind of method. So it's very tractable and it gets easy enough to do in fairly quickly and in reasonably large numbers. Now, genome-wide studies, you could never do that for a genome-wide study. There's millions of different SNPs across the whole genome that you might be curious about. You can't possibly run a separate PCR reaction for millions of different SNPs. It's just, it can't be done. You need a faster and more efficient way to genotype that many. So you also need gigantic sample sizes. So not only would you have to separately genotype millions of different variants or SNPs, you'd also have to do it in thousands or tens of thousands of people. So it's just completely impossible to ever imagine doing that with PCR. You need a genome-wide approach. The approach that they use is called a microarray. And a microarray, kind of from the top down, if you were to look at it under a fluorescence microscope, would look something like this here. What it is essentially a glass microscope slide covered with millions and millions of small circular points. Each point contains a specific probe that's been deposited on the surface of the slide. Each probe is complementary to a given SNP and its flanking region that you might be interested in. Usually these are like pre-made. You can just buy them off the shelf and you don't have to design them yourself. And so the basic principle here is you take the DNA from your participants or your subjects and you extract the genomic DNA and break it into small fragments that are about 100 base pairs long. You can do this with sonication or you can do it enzymatically. There's a number of different ways to break up the DNA, but DNA is quite long by default, right? It's long, stringy stuff. You want to break it up into manageable chunks. And then you attach fluorescent labels to the participants, or to the subject's sample DNA. So all of these 100 base pair fragments are fluorescently labeled. Then you take your microarray, which, as I said, is covered with thousands or millions of probes that are complementary to various SNPs and their flanking regions. So here's an example of what the sequence of a probe might look like. The actual polymorphism is here. It's located here, and it could either be a C or a G. The flanking region, so that's the part that's different. And then you've got a little flanking region to either side that matches with the genome. You couldn't, obviously you couldn't just have one nucleotide. If you just had a T or a G with nothing else, then that would make no sense. You have to have the flanking region to make it specific. So the probes are made in this way, and then when you expose your fluorescently labeled DNA to these probes, either they will bind and hybridize or they won't. So imagine here you've got a participant that you've taken their DNA and fluorescently labeled, and it manages to bind to this probe because it is exactly complementary to the sequence of the probe, including the variant site here, this C that's right in the middle. The person there has an A in that position. So it binds perfectly, and when you wash the probe off, everything that hasn't bound washes away, so you're left with only what's bound. So what you will see in the position of that probe is a fluorescent signal, because the fluorescent signal has been attached to the fragments of participant or sample DNA, and it's stuck there, so you'll see that circle light up on the microarray. If the person is negative for an allele, the binding won't be successful, because while these probes will match fairly well with the oligos or the with the sheared sample DNA, it won't match perfectly, and that's enough under the chemistry that they've established to prevent it from hybridizing. So you won't see a signal from that particular probe. And then you can go through and scan these slides and go probe by probe and say, okay, so at this probe, the person had the C or the A allele and it bound, but in the other one they didn't. So they're homozygous for the A allele. Or if they had both probes bind, you could say they were heterozygous. Or if they only had the probe for the minor allele bind, you could say they were homozygous for the minor allele. You can see how you can kind of go through and probe by probe genotype many, many different SNPs on a fairly large scale. Now, this is still the main method that's used for genome-wide studies. The reasons are many, but it has to do with the fact that these are quick and easy to do. On a chemical level, really all you need to worry about is getting the subject's DNA, shearing it up into conveniently sized pieces, and then exposing it to the array. This is a fairly simple process, all things considered, and the arrays themselves are not terribly expensive either, and then you can just scan them in a machine that scans them, and then you're done. So this is comparatively simple to pull off, and the advantage, of course, then is you can have giant sample sizes that enable you to have the statistical power you need to detect variants that affect mental health or neurological diseases, even at a fairly low effect size. This is also the method that are used by those kind of genotyping websites like 23andMe and Ancestry.ca. If you send your DNA into those people, they'll use custom-designed microarrays to genotype you for a whole bunch of things that they've determined are interesting or relevant for whatever service it is that they're providing. So this is fine, actually, for genotyping known variants. These methods work very nicely, and they're, despite being comparatively old technology, they're still used widely in academia and in private industry. So they don't capture every variant, but because of linkage disequilibrium and the Human HapMap project and others, we know which variants tend to be in LD with the index variants. So even if you don't genotype every single variant, if you genotype variants that are in LD with the other ones, you can tell, or you can sort of impute which ones are missing among those that you didn't measure. Well, I'll get into this later. This is something I want to talk about a bit more in a future lecture. But the point is that you can gain a lot of information for this, even though it's only sampling a small subset of the variants in the genome. Thanks to linkage disequilibrium and imputation, you can infer what's happening in a whole bunch of other ones just because they're linked. Now, that's all well and good, so it's a very useful technique in many ways. However, there are some major limitations. Arrays can't identify de novo mutations. De novo mutations are ones that occur spontaneously, and they're typically unique to a single individual. So it's impossible to design arrays that can figure these out, because arrays can only be built on known variants. You have biologists and bioinformaticists who go through all of the known human variants, and they'll design probes for every single one of them, and variants that they don't know about, they can't design a probe for. So it's impossible for arrays to identify new mutations. And that's actually pretty relevant, because if a given condition is caused by de novo mutations, or if de novo mutations play a big role in it, all of that would be missing. So you would infer that there was a strong genetic component to something based on twin studies and based on pedigree analysis, but you might miss it if the mutation isn't part of the array. So this is fairly relevant. Arrays can also cannot easily do phasing. We'll talk about phasing more in the future, but essentially what this means is determining whether a given variant that you've genotyped is located on either the paternal or maternal part of the chromosome. So in order to do this, you need to know not only the variant, but the sequence that's flanking it, because you need to be able to link it to whether it came from mom or dad. We'll get into that later, but that's a fairly relevant thing too. It's also very hard, though not always impossible, to identify deletions and insertions and copy number variations, because these are structural variants that change the way the DNA is organized. And microarrays are not sensitive to how the DNA is organized. Your DNA could be, it breaks everything down into 100 base pair chunks, and they just bind or don't bind. The microarray has no way of telling what order they came in on your actual genome. All they can tell is whether something is there or isn't. So if things have gotten mixed up or out of order in your DNA, the microarray is completely unable to tell that. So I'm just going to pop over back to the chat. We've got another question there. Could you clarify what is meant by genotyping variants that are in LD? I think I missed it. Yes. So LD means linkage disequilibrium. And the basic idea is this. During crossing over, as part of meiosis, as I talked about, the different chromosomes exchange pieces that are homologous to one another. And so as the germ cells are being produced, chunks of DNA from the homologous chromosomes are being switched around to create novel combinations that don't exist in the parental tissue, right? Now, this crossing over happens at the level of haplotypes. These are long chunks of DNA that move all at once. They move in big blocks. It's not like you're chopping the DNA up into little tiny pieces and moving all the pieces around. The pieces that move are in big chunks. And these chunks travel together. So if you have a SNP that's in the middle of this chunk, it's going to travel together with all of the SNPs that are also on the chunk with it. So really, all you need to do is just genotype one SNP inside of each of those haplotype blocks or inside of each of one of those chunks, and you can tell with a fair degree of confidence what the SNP will be in all the other ones, because you know that those all travel together, and they kind of don't tend to separate too often. They do somewhat, but they don't tend to separate as commonly. And so that is what linkage disequilibrium means. I'm going to tell you more about that in a future lecture and really try to emphasize that, because it's a fairly important part of the overall structure of the genome. But that's the basic idea, right? Is that a lot of SNPs travel together in a group and don't tend to separate very often. So you don't need to look at every single one of them. You can just look at one and use what you know about LD, linkage disequilibrium, to kind of infer what the other ones probably are. It's a bit like, this is like maybe not a great analogy, but it's kind of like, if you're at the like imagine you're at a bar or a restaurant with your friends or a club or whatever it is that people do these days, and you're there with your friends, so you're all traveling together, right? You're all going to the bathroom together or going to the dance bar together, like whatever it is that people do when they go to clubs. Like, you all travel together. So if somebody knows that you all travel together, they don't need to ask where every single one of you is. They just need to ask where one of you is, and the odds are pretty good that you're all in the same place. So it's that kind of idea, right? The variants tend to move in chunks that are based on how close they are to each other. That's a whole, like I said, that's a whole topic that's worth getting into, and we will. But for now, that's all you really need to know. So let's get into DNA sequencing, which I would describe as the Swiss Army knife of genomics and epigenomics. And the reason is that, much like PCR and the concept of hybridization is exploited in many, many different assays and approaches, next generation sequencing has led to this explosion of methods that use it in very creative ways to get all kinds of interesting data about the genome and about the epigenome. It's not, the name would suggest that the only thing it's really good for is either genotyping people or assembling new genomes. You remember the Human Genome Project, which I guess at this point probably happened like before many of you were born. But that was originally what DNA sequencing was good for. Was you would take DNA and sequence it and put it together, and you would have a map of the genome of a given species. Now that that's already been done for most species, you can use it in all kinds of other creative ways that we'll get into. So I call it the Swiss Army knife of genomics and epigenomics because it can do so many things depending on what you feed into it. And you'll just have to wait and see what those are. They're really quite clever and creative. But I think before we get into that, we have to learn how it works itself. And then once we've learned that and you've had some time to kind of digest how DNA sequencing works, then we can go in and look at how it's being used in all kinds of clever and creative ways. Okay, so let me go forward. So, wait, did I? Yeah. So the many of the methods that are central to molecular biology exploit at some level the mechanisms that are already existing in nature to do DNA synthesis. Now, of course, DNA can be copied perfectly many, many times over, and this tendency to be able to be copied perfectly leads to all kinds of tricks. PCR, in addition to using the trick of hybridization, also uses the trick of DNA synthesis, because you have DNA polymerase in your PCR reaction that's actually building the new strand of DNA and copying it. Cloning also uses synthesis, and of course DNA sequencing. So the way it works, of course, is that you have a piece of double-stranded DNA. It's unzipped from itself, and one single strand goes one way and the other single strand goes the other way. And then because of the fact that base pairing is complementary, if you have one single strand of DNA, you can tell with complete accuracy what the other strand should be. So if you have an A, you know that a T goes on the opposite side. If you have a T, you know that an A binds with it. If you have a G, you know that a C binds with it, and so on and so forth. So you can use, this is how DNA is copied. This is how it's possible to replicate DNA flawlessly, zillions of times over. Adding each base to a growing strand of replica DNA is a separate chemical reaction. So the polymerase enzyme has to grab one of the bases, stick it in place, bind it up, and then sew it together with the strand as it's growing. Again, not worth getting into the chemistry, but every one of these bases being added to a growing replica strand is a separate chemical reaction. So, in a sense, if you're curious about the sequence of DNA that you are if you have a sequence of DNA, or a stretch of DNA, I should say, if you have a stretch of DNA and you're curious about what the sequence, there's one person, or there's one guy that knows what the sequence is, and that's the DNA polymerase enzyme. Because if you take a single strand of DNA and you mix it with a bunch of loose nucleotides, and you put in a DNA polymerase enzyme, the enzyme will go to work and it will find the complementary base for every base in your single strand, and it'll attach it. And it'll go through and it'll say, like, okay, here's a C, let me grab a G and stick it on there. Here's a T, here's an A, let me grab a T and put it on there. Here's a G, let me put a C, et cetera, et cetera. And so kind of the analogy I thought of when I was trying to explain what this would mean for our purposes, like, imagine if you could listen to the DNA polymerase work. Imagine if the DNA polymerase, every time it added a new base to the growing replica strand, shouted out what base it was adding. So it would, so looking at this picture, the polymerase is this like sort of Pac-Man looking thing. But imagine it's going along and says, oh, G, T, C, T, A. It just shouts out the name of every base it adds. And if you were listening to that and writing it down as it went, what you would know is the sequence of the template strand, because of course the template strand would just be the complement to what the DNA polymerase was adding. That's the beauty of base pairing. So that's kind of the analogy at the center of this, is that you're trying to like listen or like interrogate what the polymerase enzyme is doing as it's copying your template strand. Now, the enzyme doesn't talk, but maybe there's a way to have it report on what it's doing in some other way, and this could allow you to determine the sequence of any little piece of DNA you mix in. It happens too quickly, of course, to observe in real time. The polymerase works at a rate of 1,000 bases per second, so very quickly it would copy a template strand of DNA, and you'd never have a chance to observe it in real time. But our old pal Frederick Sanger, back in 1977, invented a clever solution to kind of circumvent this problem. So Sanger's an interesting character because he's a double Nobel Prize winner, and he won his first Nobel Prize because he figured out a method to determine the amino acid sequence of proteins. And so he did that, and I think he sequenced like vasopressin or something, maybe insulin. I can't even remember what it was, but it was some kind of important but fairly small, easy to sequence protein. And he won a Nobel Prize for that. And then his second Nobel Prize, he invented a way to sequence DNA and figure out the nucleotide sequence of DNA. And that's I would say those are pretty good accomplishments. This is an approach that he invented, and it's kind of old-fashioned now. You wouldn't tend to use it a whole lot, but the basic logic of it is still at the heart of next-gen sequencing. But this method is called Sanger sequencing quite often, in honor of our old friend Frederick Sanger. So the modern way of doing this uses these things called terminator nucleotides. And the basic idea of this is that they are they're chemically modified nucleotides that when they're added to the growing template strand, they stop the operation in its tracks. So imagine if you had, 1 out of every 10 Gs in your mixture was a terminator. What would happen if the DNA polymerase grabbed one of those terminator Gs and added it? It would stop the DNA polymerase from being able to move any further. So the strand would stop in its tracks and it couldn't be copied anymore. These terminator nucleotides are fluorescently labeled with one of 4 colors representing which base it happens to be, and you can basically mix these in at a small fraction in the polymerase reaction, and look at the results. And by based on the color of the strands you get out, you can tell what base was added last. So let me try to make that a bit more real. These terminator nucleotides that have been fluorescently labeled are mixed with the normal nucleotides. The terminators are called DD-NTPs. The regular nucleotides are DNTPs, dinucleotide triphosphates. So the regular nucleotides, if they're added, the template or the replica strand can grow freely. If the terminator nucleotides are added, the replica strand stops growing. Okay. Now, these terminator nucleotides are added in a smaller amount in a lower concentration. So maybe 9 times out of 10, when the polymerase is grabbing a nucleotide, it grabs one of the regular DNTPs. But 1 out of every 10 times, for example, it might grab one of the terminator nucleotides, and that strand that it was working on stops there in its tracks. And where it stops, this is the most important part, where it stops is fluorescently labeled with a color that matches with the base that terminator nucleotide was. So if you're going along and copying this piece of DNA and you grab a terminator nucleotide, a G, for example, and it's labeled with green fluorescence, what you would see is that strand would stop, and whatever length that strand would be would tell you how many bases along there was a G in your template sequence. So you can measure this out using gel electrophoresis. But basically, the gel electrophoresis will tell you the length of the fragment when it stopped copying, when the terminator nucleotide was added. And then by the color of the band on the gel, it will tell you what nucleotide that was. So for example, here, this particular strand stopped on the 21st nucleotide, and it picked up a G. The next time the reaction stopped was on the 22nd base, and it picked up an A. And you can kind of go down the line, and by reading the color and the position in the length of all these different terminated reactions, you can infer the sequence of the template strand. You see A, G, C, G, T, C, etc., etc. So knowing the position and the identity of all the nucleotides in your template strand, that is the sequence. Do you see what I'm saying? It's quite a clever technique. It's very sort of slow and time consuming, but the principle is quite sound, I would say. So without getting too much more into the kind of chemistry of it, what we're essentially doing is imagine if you had to figure out the spelling of a word, and you were given 2 pieces of information. You were given, number 1, the letter, and then number 2, where in the word that letter appears. So as an example, you've got G1, M5, O4, N3, C7, S8, I6, and E2. So the G is the first letter, the E is the second letter, the N is the third letter, and so on and so forth. What does that spell? Has anyone figured that out? I'll leave it to those playing along at home. But anyway, that's the idea of Sanger sequencing without, burdening you too much with the chemistry of it, although I think the chemistry is very clever. That's what you get. Basically, you have a list of nucleotides, and you have a list that tells you what order they're in, and you put them together, and that tells you the sequence. It's not quite the same as listening to the polymerase, but it's sort of a way of like recording the process as it plays out. Yes, so Nathalie writes genomics. That's correct. It is genomics. So that's the idea of it, right? Is you're taking these letters and you know what order they're in and you know what the letter is. You can put that together and figure out what the sequence is supposed to be. So it's not like listening to the polymerase, but it's a way of kind of recording it as it's happening. And then you can observe it later on after it's already played out. This method was invented, it was used for the Human Genome Project that happened at the end of the last millennium. And the Human Genome Project started in 1990 and ran for 13 years at a total cost of $2.7 billion, only, just to sequence the first human genome. And Sanger sequencing was used for most of it. Very slow technique. You're working with little chunks of DNA that are maybe a few hundred to, a few hundred base pairs long, kind of at the longest. So imagine sequencing the human genome at a rate like that's that slow. There's, billions of bases in the human genome. Imagine trying to sequence it 100 at a time from little clones that you get out of little E. coli. It's a very, very time-consuming process. That's why it cost so much money and it took so long. So it is still actually used for some applications today. It's a lot cheaper than it used to be, but it's not at all appropriate to doing genome-wide sequencing. However, it gives you an example of the idea of sequencing by synthesis. And this is the heart of next-gen sequencing as well. Synthesizing DNA gives you a window into the bases that are being added, and if you know what bases are being added, then you know the sequence of the template strand. And that's the aim of sequencing, is to figure out what the sequence is of the template strand. Next-gen sequencing uses this at a much higher rate. One of the main kind of motifs that we'll see in genomics is taking a technique that works on a small scale and figuring out a way to scale it up massively. I think that's kind of the secret of this is like of American success. Maybe if you were to think of like what makes America successful, it's not really anything other than just being able to scale stuff up on a massive level and take something that somebody could do. One person could do over the course of a week, and figuring out how to take 1,000 people and do it in an hour, that kind of thing. So next-gen sequencing is kind of a scale-up of this. It uses what's called reversible chain termination sequencing. So it uses the same idea of terminator nucleotides, but in this case, the termination is reversible. So you can have it stop the reaction and then reverse that and have it restart the reaction. It's sort of a massively parallel automated version of Sanger sequencing, made possible by the fact that in this particular case, the terminator nucleotides are reversible. So they can stop the reaction, and then you can start the reaction back up when you're ready. So it's a bit like having a way of saying to the polymerase, okay, give me, shout out what you're doing one base at a time. I'll tell you when you can stop and start. Just add one base, tell me what it was, and then you can go on to the next one. So the Illumina sequencing, or next-gen sequencing as it's called, is essentially just pauses the reaction, where in Sanger sequencing the reaction is stopped. So as I said, most of the best inventions in genomics really just come from taking existing techniques and scaling them up on a massive level. And Illumina sequencing is big science. I guess next-gen sequencing is what you really should call it, but sometimes I'll call it Illumina sequencing because Illumina is the company that sort of owns all the patents for it. These are big, fancy machines. This is one that I found on the internet. This is the University of Pittsburgh Medical Center Genome Center. These are Illumina NovaSeq 6000 machines. These are sort of state-of-the-art machines. The sequencers all look like this. They look like big photocopiers, kind of. And a setup like this with several sequencers running in parallel can do for about $1,000 what the Human Genome Project did in 13 years for almost $3 billion. So this is the level that we're operating on now, where these machines can do this on such a ridiculously large scale that it puts a several billion-dollar project that took decades or over a decade to shame. It can do it in a weekend practically. The reaction takes place in this thing called a flow cell. This is an object about the size of a glass microscope slide. It is made of glass, and you basically put your sequencing libraries into these lanes, and they'll flow out, and the reaction all takes place on these sequencing lanes. So let's get into the chemistry and the kind of the way this works on a step-by-step level. So As always, you begin with a sample. The sample could either be cells from a cell culture, or it could be a piece of tissue, or it could be DNA you got any other place. I've got these little circles here. These represent cells. Maybe they could be scrapings from the cheek, or they could just be whatever you like, right? That's the first step. The second step is you have to lyse the cells and purify the genomic DNA. Here, the DNA is represented by these gray bars. And coming right out of the cell, DNA is quite a long, complicated molecule, right? Very high molecular weight DNA is what you get. Way too big to really do anything with on this level. You have to break it up, as always, or as with the microarray technique. You have to break the DNA up into small fragments that are about 100 to 200 base pairs in length. And this can be done enzymatically. The little scissors here are just, cartoonish illustrations, but it could either be done enzymatically or it could be done with sonication by just physically shaking the DNA around until it breaks apart. Whatever the way it is, it doesn't particularly matter, but you've got to get it down to smaller lengths. And then you ligate or attach sequencing adapters. So these are specially prepared oligonucleotide, essentially probes, or things that are kind of analogous to the probes I was telling you about, that are able to bind to your little template strands, the little 100 to 200 base pair fragments that you've made. They'll bind to both ends of these. And these sequencing adapters are made to be complementary to the sort of probes, I guess you could call them, lining the flow cell. So they're complementary and they're sort of universal. So it's like putting it's literally an adapter. It takes this random piece of DNA that you don't know what the sequence of it is, and it adds onto each end of it a predictable sequence that is given to you by the manufacturer. This sequence is able to bind to the flow cell, allowing your sample DNA to stick to the flow cell and be analyzed. Now, adapters can also have inside of them a barcode component that makes every fragment of DNA uniquely identifiable. This allows you actually to mix different samples together in the same flow cell and mix up all of your samples, because as long as they're barcoded differently, you can pick that up when you're doing the analysis and kind of digitally dissect them and figure out which data came from which samples. Anyway, but this adapter ligation is a key part of it. And the adapter ligated or the adapter tagged DNA is then put into the flow cell. And the flow cell, as I mentioned, is basically looks like a microscope slide. And much like a microarray, the slide is lined or is dotted with complementary oligonucleotides, or sort of probe-like things that have been fixed onto the glass. And when your sequencing library comes in with all of these adapters ligated to it, they'll bind to the complementary probes on the flow cell, and they'll stick there. And they'll just they'll get stuck to the glass slide and kind of stick sort of straight up like this. And they'll be permanently kind of immobilized there. So you've now put in little pieces of DNA into the flow cell. You've stuck them in place so that they can be, they're sort of fixed into a given spot. And the next thing that happens after that is you do what's called cluster amplification. So the concentration of DNA you're putting in is not necessarily that high, and it's a little tough to work with that well, because these machines need more DNA to give a bigger signal. The bigger signal is easier to detect reliably. So without getting too much into how this works, essentially what cluster amplification does is it takes these little pieces of sequencing library and it amplifies them in and around where they've bound. So I wrote down here, I'm really simplifying this process because it's not really part of what you are here to learn necessarily, but it's really quite elegant. Like if you ever have any spare time, please go and just look up a video or read about in more detail how next generation sequencing works and how cluster amplification works in particular. It's really quite a marvel of engineering. So a lot of credit goes to the people who invented this. But anyhow, the way it looks like to begin with is you've got your little pieces of sample DNA, your libraries, that have bound up onto random spots on the flow cell, wherever any of them happen to stick. And because the libraries have gone in on a fairly low concentration, all of the spots where they've stuck are very small, like physically not big, because maybe only one piece of DNA actually stuck in that spot. After cluster amplification, the physical number of copies in each of those spots increases, and therefore the physical size of the spot also increases. The fact that these spots become bigger makes them easier to detect. Each one of these spots is basically like it's detected by a camera. So the more pixels you have for every spot, the easier it is for the camera to tell what's happening there. Now, a real-life flow cell has billions of these little spots or these clusters on the surface. I've given you maybe like 10 or so to look at, but really there's billions of these that are all there being analyzed. Next begins the sequencing cycle, and here's where the sequencing by synthesis comes in. So now, just to summarize, we have got a bunch of you've basically taken the DNA from your sample, you've broken it up into small pieces, you've stuck all those pieces into little spots on the flow cell. They're stuck there, surrounded by copies of themselves, so it's a nice big sample to look at. And you're trying to figure out, okay, for each of those little spots, what is the sequence in that one little spot? And the logic of this is the same as Sanger sequencing. If you have let's say you're looking at this first position here, and it's an A. How do you find out if it's an A? The way you find out it's an A is by putting in nucleotides and seeing which ones bind. The one that will bind is a T, because T base pairs with A. So in this particular spot, a whole bunch of T's are going to bind. Each, just by pure kind of Brownian motion, they'll just bounce around until they find the right place, and then they'll stick there. And after it's bound, the reaction is paused because each of these is a reversible terminator. So one nucleotide will bind, and then everything will stop. So in the sequencing cycle, you expose the flow cell to these fluorescently labeled reversible terminator probes. They float around. Eventually, they all bind. Then nothing happens. Then it stops. Next, what you do is you wash off all the ones that haven't bound. You've run, essentially buffer or water over it. All the ones that haven't bound are flushed away, and only the probes that have bound remain stuck because they're base paired onto your library or onto your template DNA. Then, using a laser, you image the surface of the flow cell, and because each of these 4 nucleotides is labeled with a specific color or a specific colored fluorophore, when you scan this with a laser, you can look at the fluorescence that's returned by each of the different spots or each of the different clusters on the flow cell and look at the color. And by looking at the color, you can tell what base was just added. So in this case, for this particular cluster, we just added a T, and T is colored red. So when you expose this to a laser and record the signal on a camera, you'll see red come back, and that tells you that the first base that stuck in this cluster was complementary to T, and that means it was an A. So that gives you your first answer. Then you can chemically reverse the termination process. That means that now this T that was added no longer stops the reaction. It lets one more base join in after it. So then you would return back to the beginning of the cycle, all the way back to here. So after the T is bound, what comes next? Well, whatever is complementary to T, so that would be an A. So the next base that's going to be binding is an A. So you let the A bind. When the A binds, it stops the reaction because it's a reversible terminator. Then you image it again, and what you'll see instead is the fluorescence coming from the A, not from the T. So you'll get a yellow signal, and then you say, okay, so first we had a T in this spot. Now we've got an A. That means, you see where I'm going with this. You can go through and do this down the line, and you'll get the whole sequence of the whole fragment in that particular spot. And because you've got billions of spots on the flow cell, you'll get a large chunk of the genome sequenced. So basically, as this cycle progresses, what you're seeing through the camera is a sequence or a pattern of colored signals after every cycle, and then you can infer from these fluorescent signals what the bases might be. So if you see a red first, that means you added a T. If the next signal is yellow, that means you added an A. The next 2 signals after that are both blue. That means you added 2 C's. And then the final signal is a green. That means you added a G. And then so you can say, okay, well, put this all together. What did we have in this area? We had TACCG. And of course, complementary to that is ATGGC. So that's how you tell the sequence from adding these probes one at a time, or adding these fluorescently labeled reversible terminator probes one at a time. You can tell one at a time what the sequence of the DNA is in every cluster. Now, remember, every cluster, there's billions of these clusters on a flow cell, and every cluster yields about 100 to 300 base pairs worth of sequencing data, depending on the exact way you run the reaction. So you can get, billions of clusters times, let's say, 300 base pairs. That gives you a very large chunk of the genome all at once. The next step after that is alignment. So what you get from this process are what are called sequencing reads. So every little cluster on the flow cell maybe has 100 to 300 base pairs on it. I'm giving you a shortened example, of course. But what you get back from the camera is a set of nucleotides, right? So you might get a G, C, T, et cetera, et cetera. Each of these sets is a read, and it comes from a random piece of the genome. Remember, we broke the genome up into random chunks through enzymes or through sonication. So you've broken it up into completely random chunks, and you sequence all of the random chunks. And then in the alignment step, you're taking all those random chunks, and you're trying to figure out where do they fit on the reference genome. Here's the reference genome. This is what, let's say this is the human genome. Here it is, and you get a piece that matches here. So this goes, you can map this to here. This next fragment, or this next read, overlaps a little bit. This part matches with the first read, but this part matches over here. And then the next read comes in, and it fits over here. And you can kind of just go through, and by matching the reads up to the reference genome one at a time, you can tell where they belong in the reference genome and what genes that they're part of or whatever else. When you do the first assignment, I kind of give you a very basic exercise using Control F in the in a sort of fake reference genome, and I'm doing this just to show you okay, if you have a sequence of DNA, you can find where it belongs in the reference genome just by searching through and looking for a match. This is essentially what the alignment process is doing, although it's much more complicated than that, because of course the alignment has to contend with variance. Not always is it going to match. Maybe the reference genome has an A here, but in all of your reads, there's a T instead. That would be a polymorphism, and the algorithm has to be able to handle that. But basically, this is the way it works, is you're taking these little pieces and trying to match them up with where they go in the reference genome. And then after that, you can look and see how your samples differ from the reference genome, and that's how you are able to genotype people. Okay, so somebody asks, does step 7 always happen in order? So the first nucleotide binds, then the next, or then the next. No, so it always happens in order. That's kind of the beauty of it, because the termination step. Because DNA is synthesized from in order from 5' to 3', there's a directionality to it. So the first base always has to start in the first position, and then the next base has to be added to the next position after that, going in order. It's not possible to just add them bit by bit, because the polymerase enzyme essentially wants to stitch together a complementary strand. So it always goes in order. That's kind of the secret of it, right? The enzyme has figured out how to put these sequences together in order. We're just exploiting that ability to figure out what the sequence is. So just a few words about data quality in these experiments. This next generation sequencing, or NGS, is able to generate vast quantities of data. You can sequence an entire human genome this way. You could theoretically do this by just having a few reads covering each part. Like what I've shown here, this part of the reference genome here, for example, is covered by 1, 2, 3, 4 reads. So that's comparatively low coverage, actually, but technically this would be enough. You know what's going on there. If you have 4 reads, you've got 4 chances to genotype every position. So that's not really that great, especially if somebody's heterozygous. You might see 2 reads that have a T and 2 reads that have an A. That would mean they're heterozygous for a variant there. Really, the more reads you have covering every part of the genome, it's better, because you can be more confident about the data you get. There is an error rate to this method between 0.1 and 1%. So you need a lot of reads to kind of compensate for that, to make sure you're actually sure about what you're seeing. The kind of the 2 most important parameters here are coverage and depth. Coverage means how much of the genome is covered by your data. So across the whole genome, how many parts do you have any kind of data for at all? And then depth refers to, for any given part of the genome, how many reads do you have stacked up on top of that, and thus how confident are you? It's kind of like sample size in a sense. So I gave you this example of snowfall accumulation to show how the two are kind of related, because you can talk about snowfall in both coverage and depth, right? There are parts of the yard, like at least at the beginning, that are covered more because they are they're insulated more. They're part of the ground, or they've got mulch or grass on them. And then there's parts that are covered less because there's pavement there. And depth is, of course, how deep the snow is. So like on branches, it gets to be quite deep because there's almost no heat stored in those. And in other parts, there's less depth because some of the snow is going to melt as it falls. So, genomics data can be described in the same way, coverage and depth. And so here it is represented visually. Each of these little gray bars represents a sequencing read. And this diagram here represents a gene, just some random gene in the genome. And here is what it would look like if you had high coverage and low depth. Every part of this gene is covered by at least one read. Some parts right here where the reads overlapped have two reads covering them. But basically the coverage is about 1x. Any given spot here has on average one read covering it. Maybe it's 1.2x, you could say. Here's an example of high depth but low coverage. The parts where you have data, there's a lot of reads stacked on top of each other. So you are very confident where you have data, you're very confident in what you're looking at because you've got so many different samples or so many different so many different reads telling you what's happening there. So if you're making a genotyping call, you're very confident in it. However, what's happening over here, you have no idea because that's not where you have no reads over there. You have no information from parts of the genome that aren't covered, you see. And of course, optimally, you would have high depth and high coverage. This means the entire genome is blanketed with a nice, thick, heavy coat of reads, so that for any given part, you are looking at maybe, say, 50 reads all stacked on top. You have 50 chances to tell what the genotype is there, and thus any type of error that you might have, you can be, errors won't affect things too much because errors happen at a rate of maybe 0.1% or 1% at the highest. So if you have 50 reads and you see one that doesn't match anything, you would know that's an error. You'd be quite confident. So the more samples you have and the more reads you have, the more confident you are. And of course here, they're covering the entire area, meaning the level of coverage is also quite high. So those are the basic parameters to look at, coverage and depth, and ideally you want high depth and high coverage. That's the best scenario. So it is possible, as I said, for there to be errors. Incorrect base calls is what it means. It's when the machine tells you that a given thing, or that there's a given base in a certain position and it's not actually there. This can happen once in a while. The machine has to make decisions based on noisy data that are coming in through its fluorescence detection system. So it's not flawless. Depending on the quality, it could either be between 0.1 and 1%. At a 1% error rate, that's fairly high. If you were sequencing the genome at a low depth, 1x, you would never know for any given read whether you were looking at a mistake or a real SNP, right? If you see a T, like let's say right here, if you see a T there and it's supposed to be, the reference genome says it's supposed to be an A, how do you know that it's a SNP or an error? You have no way of knowing with one read. You need a lot of depth to be able to tell for sure, because if there's, let's say you're looking at this area and you see, let's say, okay, right here, there's supposed to be a G, but here you pick up an A on this one read. Now, one read telling you a base that's different from every other read you get, that read probably just has a mistake in it. So you need a lot of reads to be able to account for this. Heterozygous variants, of course, are more difficult to detect. If you have a homozygous SNP, then every read will show that SNP, and you would need, whatever number of reads you have. And if you see every single read shows you that variant, you're pretty sure the person has it and they're homozygous. If they're heterozygous, that means half the reads are going to show up on average with one variant, and the other half are going to show up with the other variant. And of course, that makes them more difficult to detect because maybe there's an error, like a 1% error on, say, 50 reads, pretty easy to manage. But if you're looking at, half the reads are one variant and half the reads are another variant, a 1% error is going to start to look a bit bigger because you're really looking at half as many reads. So again, it's just a matter of like how much detail do you think you're going to be able to detect? So another question up in the chat. Coverage is determined based on reads horizontally, i.e. how widespread is the data, and depth looks at the reads from top to bottom. Yes, exactly. Yeah. So, and the two are related, right? If you sequence, like the more sequencing you do, the more the coverage expands and the more the depth expands as well, because you're just getting more and more data. Next-gen sequencing can detect indels. These are insertions and deletions. These are, I think I mentioned this already, and we'll get into this a little bit later. But like I said, microarrays can't really tell if pieces of the DNA are missing or have been rearranged because it has no order. Like it can't put the pieces in order. All it can tell you is if something is there or not. Indels are insertions or deletions that are sized between 1 to 100,000 base pairs. And SNPs always happen in the same place between individuals, so you don't even need to know where they are. You just need to know whether a person has it or not. But indels can be kind of random between people, so you really have to do sequencing to tell where these are. And as we'll see, insertions and deletions and repeat expansions might be fairly important for certain kinds of mental conditions. For example, autism spectrum disorders, it looks like repeat expansion, and that's a type of insertion, is a fairly important component. So microarrays can't tell this, but sequencing-based methods can. Now, just as we're kind of closing off here, I kind of gave you the story about next-gen sequencing as it relates to sequencing genomes and detecting variants. This is called whole genome sequencing, WGS. That's only one thing you can do, though. That's sort of why it was invented, but researchers have figured out all kinds of other clever things to do with it. For example, there's what's called exome sequencing. Here, you basically select out just the exons from genes and sequence those. Exons only make up about 1% of the genome, and they're protein coding. So if anything goes wrong with an exon, it's going to have a very big effect. And so you're getting a lot of bang for your buck here because the effect of mutations to exons is very high typically, and exons only make up about 1% of the genome. So you're paying for less sequence. The amount of depth and coverage you have, instead of having to have it spread out across the whole genome, you can concentrate it just on the parts that you really think are important, the exons. So exome sequencing is a way to get sequencing quality data on a small subset of the genome that you're curious about. There's also lots of other methods like ChIP-seq and ATAC-seq and whole genome bisulfite sequencing and Hi-C. We're going to talk about all of those in the future, but right now, I mean, your brain has had enough fun learning about just what next-gen sequencing is. Later on, we'll kind of go into more about like how you can use it for clever things in epigenetics. But one thing I will mention here, just to close out, is RNA sequencing. So it's possible using the reverse transcriptase enzyme. It's an enzyme that you can get from viruses to convert RNA to DNA, to a complementary DNA sequence, a cDNA. And now that you have DNA, you can apply it to this next generation sequencing workflow. NGS needs DNA, but you can take RNA and convert it into DNA and then feed it into the sequencer. And then here, you're not necessarily looking at variants in the reads. You're not trying to tell if somebody has a mutation or not. You're just trying to tell how much of a given RNA molecule they had. Because gene expression is sort of related to how many copies of a messenger RNA are produced. So if a person has a lot of mRNA, it means that they're expressing that gene at a fairly high level. And if they don't have very much, they're expressing it at a fairly low level. So you take the RNA, you use reverse transcriptase to convert it to DNA, and then you put that DNA into the sequencer. And here, what I was telling you about coverage and depth really more specifically, is not really so much a quality control measure here, but it's actually a quantitative measure. Because the more copies of the RNA you had in your original sample, the more copies of the cDNA it will produce. The more copies of the cDNA it produces, the more reads show up over that gene in your data set. So here, each of these bars represents a sequencing read, and gene A is expressed in a very high level, so you've got tons of reads stacked up over gene A, and gene B is expressed at a fairly low level, so you have a smaller number of reads stacked up over gene B. And by using this logic of like converting sequencing depth into a quantitative measure of how much of that transcript or how much of that RNA was in your sample to begin with, you're actually able to profile gene expression for every single gene in the genome all at once. So you can look at all 20,000 genes all at the same time. And that also includes all the different microRNAs and non-coding RNAs that are in the mix as well. So this is a way to use next-gen sequencing to get quantitative data. More reads equals more expression. And that logic will apply in other places like ATAC-seq as well as ChIP-seq. So so keep that in mind, that this is another way you can use this technology. And this is why every university out there is trying to build these sequencing centers, is because they're just so obscenely useful for getting genome-wide data from all different types of things. And RNA-seq, I should point out, is also a lot cheaper than whole genome sequencing, because RNA sequencing is kind of like a type of exome sequencing. RNA is made of exons, or like mature mRNA is made of exons. So you're only looking at a very small part of the genome to get this kind of data. And then so the last thing I'll say is that sequencing has become a lot cheaper in recent years. The original Human Genome Project cost almost $3 billion. As time has gone on, it's declined markedly as new techniques and technologies are invented. Right around 2015, there was a nice inflection point when the Illumina, I think 10x machine was invented or released. And now, for about $1,000, you can sequence an entire human genome, compared to the cost of like $3 billion originally. This is happening at a rate much faster than Moore's law, which is the law that dictates the rate at which semiconductors shrink in microprocessors. So this has led to kind of the techno-optimist idea that someday everyone's going to have their genome sequenced, and this is going to lead to a lot of improvements in the way medical care is administered. We haven't seen that yet, and I don't even know if you would really want your DNA sequenced and filed away somewhere. So we'll leave it there for now. But this is just kind of a hint of the technology. This is stuff that, when I first got into science around 2008. So this is sort of, so around here is where I started, right at this kind of inflection point. And between then and now, look at how much cheaper things have gotten. It used to be something that would have cost millions of dollars. Now it can be done for thousands. So it's a pretty nice time to be alive, I would say. Kiana writes, wow, I didn't know that RNA quality or quantity related to gene expression. Exactly, yes. RNA quantity relates to gene expression. The more copies of an mRNA you have, the higher the level of gene expression there is. That's kind of how gene expression is able to be quantitative, right? If your cell needs a lot of a given gene, it's going to produce tons and tons of mRNAs for that gene. If your cell doesn't need very much of it, it's going to produce very few copies. So you just have to count the number of copies there are in a cell, and that tells you how much of the gene is being expressed. It's very elegant. Anyways, folks, so we're out of time here, and I just managed to finish the lecture. Managed to just finish the lecture just in time. So we'll leave it there. As always, if you have any questions, feel free to email me. We'll have office hours tomorrow over Zoom. Feel free to pop in if you want to talk about whatever. I just hang out and basically just work on stuff until people come in. So it's pretty nice place to hang out. And so we'll leave it there, folks. This is probably going to be one of the more technical lectures. So if you found this to be a little bit overwhelming or a bit confusing, just follow, try to follow through it a few more times. Try to look up other content online if you find that my explanation isn't ideal, or just ask me about it. But if it's not your cup of tea, stay tuned, because there's going to be more stuff that's like probably a lot closer to what you are, interested in coming up later. So with that, I'll wish you all a good rest of your day, and we'll see you next time.